Next, we have a live presentation. Now, I will say I did make some fun cards. So we have the 2 minutes remaining blowfly. We've got the 1 minute remaining horsefly. And we have the time flies, you're done, pagoded. So just look out for those while we're doing this. And so next, please welcome. We've got Riley Hoffman giving a presentation, Blowflies Under the Lens, Forensic DNA Retrieval Post-ESEM. All right, welcome. Thank you.
Thank you for being here. My name is Riley, and today I'm going to be presenting my research that I actually got my master's degree with at the UC Davis Forensic Science graduate program. So to begin with, caliphorids, known as blowflies, are some forensically important species. And this is due to the fact that they do have a wide geographic distribution. They are some of the first eggs or flies to arrive and lay eggs on carrion. And their larval development does help us determine PMI or postmortem interval. However, larvae are difficult to identify. So according to the organization Scientific Area Committee, it is best for them to be reared out into adults. However, this takes time and it kind of scares away untrained CSIs from collecting larvae at the scenes. So with this, I have my high resolution images for scanning electron microscopy and DNA barcoding as standard ideas that have been used within forensics and within entomology. So my research application was to couple both ESEM and DNA barcoding to reduce the need for identification through rearing to have greater accessibility to forensic entomology on the forensic side. And here I have my lovely model species, the Chrysomya rufifasces, and I use them because they're super easy to identify by the eye. As I said, I'm a forensic scientist trained, but more of an entomology enthusiast at the moment. So I wanted something that I could very easily identify. And also, I was very lucky to get mine from Texas A&M. So looking at all the options that Texas A&M gave me to pick from, this was the one that actually I could find the best published genome for, and the one I went with ultimately. And my research questions were to, one, see if I could take these images without disturbing the insect itself. After doing those images, I wanted to make sure I could get the DNA out, and then finally use that DNA to identify the specimens. So here I just have my overall workflow. I apologize, it's been a long day. So I started out with 90 larvae, and I had them in 3 groups: unaltered, dried, and dried and imaged. For my drying process, I mixed a 50-50 mixture of ethanol and acetone together, and then I diluted that down with DI water. I then had those dilutions at 70, 80, 90%, and then 100. Each of these insects was placed in those dilutions for 15 minutes before going on to the next step. After I had my drying treatments of those 30 individuals, I then had my extraction methods down to 10 individuals. So I had whole, pierced with a fine insect pin, and homogenized larvae. After my extraction was complete, I quantified using qubit fluorometry. I then amplified using PCR, looking at the CUI1 gene. And then I tried to visualize my fragments using UV visualization and agarose gels. I once again quantified using fluorometry. I then had to purify my PCR product before sending it off for Sanger sequencing. And then I looked to see if I could actually use my sequences for identification. And here are my lovely images, if I do say so myself, of my larvae. So first we can see the one that had been through the drying process and a larvae that had not been through the drying process. This is done because environmental scanning electron microscopy does not require larvae to be dried. However, previous literature mostly had published using SEMs, so I just wanted to keep consistent with what I knew worked. Additionally, having that moisture be present can cause blurriness, or what we call noise, in photos, and going with the idea that these photos could potentially be put into a court. I wanted to make sure that I was doing what I could to give us the best possible photo, and doing this, I also wanted to confirm that I was not changing the external morphology through my drying processes. So I first have my ANOVA here to show if anything was significant via my extraction process or my preparation for imaging just out of my normal DNA extraction. And we can see that nothing was significant here, which I was really excited about initially. And here I just have show what I did for my PCR primers. I did test all of my primers, and I ended up going with primer pair 1. This was due to the fact that it showed the most consistency in being able to see these bands in the UV visualization. And next was my analysis of variance for my post-PCR. And here we can see that I do have something significant. I had that my extraction method was significant. And so my question was, well, why did it show significance here and not initially in my extraction processes? Well, I can think of a couple reasons. First off, my larvae were blood fed by bovine blood, and I was curious to the fact that potentially I was getting some bovine blood. I also could have had fragmented DNA, which would not have been able to successfully amplify. And then finally, I also considered the fact that I could have just had a lot of extracellular DNA. Due to the fact that qubit is not specific to Diptera DNA, I definitely was just getting any DNA that was there for my readings. So to remedy this in the future, I would love to see some shotgun sequencing to see what is actually in my samples. I also think that doing primer-specific qPCR to see how much Diptera DNA I was actually extracting at all of those points. And finally, I think that nested PCR could really be successfully in getting that beautiful and consistent amplification of my fragments. But here was the exciting part. I was still able to get readings for 6 of my samples. And we can see that compared to published sequences of Chrysomya rufophaces, I was able to successfully identify them using the fragments that I got out. However, it's important to note that when you look at the other Chrysomya groups, I have very similar percentages. So my percentages range from 1.4 to 5.1%. And it's worth noting that most people like to have a species delineation of 3% and higher. So I'm thinking that the best course of action would be adding another gene like the ITS2 gene. It is also worth noting that there has been some studies that find Chrysomya have as little as 0.5% delineation. So that definitely causes me to want to have more genes in the future. And my conclusions came in as having Chrysomya rufophaces larvae can in fact be dried for ESEM imaging, that the DNA can be extracted from the larvae in a minimally to non-destructive manner, and that the DNA testing and SEM imaging are best when completed in an academic entomology lab setting due to the instrumentation that I used in this setting. We'd want to make sure that we'd have that. And then finally, that that was a slip. ESEM and DNA barcoding can reduce rearing time. And going back to that closeness of all of my results in seeing that species delineation, I think this is the part where I can really say, yeah, there's a lot of things that DNA can do, but this is the part that I know I'm preaching to the choir here, but usually when I go to conferences, I say that's when you want your expert witness of your forensic entomologist to look at what species are normally in this location. What is the time of year? They might be doing resource partitioning. We know best of what could possibly be there. And it would just also speak to the knowledge of why that fly is importantly found there and saying, yes, the DNA is correct, even with that closeness that we're seeing. For future research, I think it would be best to look at protocols for first and second instars to decide what would be best for processing them and not only having that third instar larvae that I used. And finally, I would really love to see a nationwide sample set of Chrysomya rufophaces to really see that variability that is within the gene, within that species, so that we can start looking at how close it is to sister taxa as well. And finally, I would like to acknowledge my advisor, Nicholas Miller. He was great. He answered every question I had. Also, I have Illinois Tech up here, but then on the program, it says Northeastern. He just made the switch, so it's kind of both places. My committee members who were wonderful in helping me get me through my master's degree. I'd also like to thank the Bond Lab at UC Davis. They allowed me to use their really fancy ESEM, and the Tyrone Lab and Tyrone's lovely grad students who took the time to send me my larvae and answer all of my questions about what they were being fed. And finally, I would like to acknowledge my lab mates, none of which are entomologists, but did begrudgingly have to watch me grind up a larvae sometimes at 8 in the morning, which they did not love. And finally, my larvae whose sacrifice made it possible for me to do my research. I am working to get my research published in the Journal of Forensic Science, trying to encourage people once again that forensic entomology is important. Again, I don't need to convince you, I'm aware, but sometimes the forensics people kind of think of it as obsolete. The people who aren't as open-minded as all of the lovely people here. And I worked pretty hard to have my preprint up. So if you want to take a look at that, you're more than welcome to. And I would also love to have conversations with you afterwards. I have my business card where I also have a QR code to my preprint as well. Thank you. Questions? No?
We do have.
Oh.
I've got one.
Okay. Yeah.
Why did you choose that section of the CO1?
Oh yes, so the reason I did the CO1 is because I was not getting consistency in the rest of my slides. I had surprise slides. Hold on, surprise slides. Yeah, so you can see that the consistency and brightness was not always there. I had like 2 primers not at all show up. And then primer 2, it does look good there. And then I started doing primer 1 versus primer 2 testing. And I even tried to combine my front primer 1 and my front primer 2 to have a bigger section. I do think that that is when having nested PCR would be really beneficial. Unfortunately, it was just outside my capability of the time. Yes.
The SEM pictures of the dried and undried larvae look pretty good. Like the wet ones look good too. So are you
my project changed slightly for having the SEM versus ESEM. So I only had access to the ESEM. I thought initially I might be able to wrangle some access at the Sacramento County DA lab and use their SEM. So that's where that comparison is going to go on. And I asked really, really nicely. They had 3 SEMs at the time, 2 broke and only 1 was working. And they said, please don't touch our stuff. And then they were also replacing 1 at the time too. A lot of crime labs are now switching to SEMs that are very specific to only looking at gunshot residue and actually wouldn't even be capable of going down to the frequency we would need to have images without completely destroying our larvae.
Well, thank you very much.
Thank you.